All right, this is part 5. At this point, we have 4 modules left in this chapter, and I don't know, there's not really a logical chunking of this, so I'm just going to do 2 and 2, just to make it in our time frame. So the first, we're going to start on 911, page 334, and we're going to talk about types of muscle contractions. There are 2 types, isotonic and isometric. And so what I want you to think about is if you are standing in the classroom and you are lifting something heavy, let's say we have, we're lifting a cadaver, I don't know. So we're lifting a cadaver. So think about all the muscles that are contracting when you lift it, when you put it back down, and just standing there. And so we're going to talk about all types of contractions using that example. Maybe a less disturbing example would be if you're at the gym, you're standing and you're actually lifting weights. All right, so an isotonic contraction, you've probably seen the prefix iso before in maybe your biology or chemistry class. We have isomeres, we have isotopes. That iso prefix means equal, and the tonic refers to tension. So this is when tension rises and the skeletal muscle length change. So that's the key, is that the length of the muscle changes. So we're going to use the example of lifting an object. There's 2 types of isotonic contractions, concentric and eccentric. The concentric contraction, essentially concentric is going to shorten the muscle and eccentric is going to lengthen the muscle. So again, isotonic, the change in the length of the muscle. So the muscle tension rises until it exceeds the load. So the muscle shortens, but the tension remains constant. So when you're picking up the cadaver or you're picking up the weights, that's a concentric isotonic contraction. And this is on page 334. This is just showing you what's happening in terms of muscle tension versus time, the amount of the load, the maximum tension, the muscle relaxes. So that kind of is the red line is kind of what we've been talking about, the contraction and relaxation. The blue line is new though. This is showing, oops, sorry, that's my alarm. The blue line is showing the length of the muscle. So at this green point is where the contraction begins, and we see our, our constant contraction, and then this maximum tension produced, you can see that's where the muscle shortens. That's this dip here in the blue line. And this is the speed that speed kind of decreases as the load increases. The other type is the eccentric contraction, and this is when your muscle is actually lengthening or elongating. So if we were putting the cadaver down, or we were returning our dumbbell, we've contracted our biceps, and now we're returning them down, you're going to lengthen the muscle. When the contraction ends, the load stretches the muscle until the muscle tears, the tension breaks, or elastic recoil opposes the load. So this is not something that can be sustained. So for this one, you can see that this red line looks very similar, but as far as muscle length, as the contraction begins, that muscle length increases and continues to increase until, a stop point. The muscle length does not change in isometric. I feel like I'm missing a slide here. Yeah, so isometric, this should be the isometric contraction. Metric referring to measures, but the muscle length does not change in an isometric contraction. And the tension never exceeds the load. And so the example that we're using, we used isotonic to lift and lower the cadaver, but the muscles making you stand there in the gym or in the lab that would be isometric. The length, and the difference here is this blue line. The length of the muscle doesn't change. It's just kind of stabilizing. All right, the second section here is about ATP. And I thought this would be a good place to kind of, 912 would be a good place to kind of end, because you might have to kind of go back and review your biology and review your cellular respiration to sort of remember all of the chemistry of it. All right, so ATP, sources of ATP in the muscles. There's a whole heck of a lot of mitochondria and muscle fibers. So we do have cellular respiration that's occurring. You remember that your glycolysis is going to produce 2 ATPs. So not huge, but it's still a source of ATPs. And this process breaks glucose, that 6-carbon glucose, down to 2 3-carbon pyruvates, or you might have called it pyruvic acid, same molecule. Aerobic metabolism or aerobic respiration. This is the majority of your ATP. So it produces, and this number varies, but overall for the aerobic, it's about 15 for each pyruvate. You might have come up with an overall number of like 30, 32 when you talked in your biology class about all of cellular respiration, so that does include glycolysis as well. So this is just an overview. This probably looks very similar to what you've done in biology. Here's our glycolysis stage, taking that 6-carbon glucose and bringing it down to a pyruvate, getting ATP out of it. Then your aerobic, so that pyruvate enters a citric acid cycle. We lose some of that carbon and carbon dioxide. Those excess hydrogen ions that come off during this cycle go to your electron transport chain and drive the production of ATP and H2O. Most energy is stored as glycogen. That's up to 1.5% of your muscle weight. Glycogen is a good source to break down and start freeing ATP up. There are free ATPs in the cells, but these are only minimal, supporting only about 10 muscle twitches. Creatine phosphate can break down and supply energy for about 15 seconds. The creatine is the carbon base and it's assembled from amino acids. You may have seen like, not energy drinks, protein powders and stuff that claim to have excess creatine phosphate. That's why, because it does supply so much energy. Glycolysis, anaerobic, that's an anaerobic process, and then you have aerobic metabolism as well. And this just kind of gives you numbers. This isn't really, I mean, it's not critical that you memorize these numbers, but just kind of compare one source to the next. So a muscle at rest does not demand a whole lot of ATP, that makes sense. But the mitochondria are still producing ATP, and so there's a surplus. The fatty acids and glucose that are absorbed from the bloodstream, and here you can see the visual here, fatty acids, glucose, they are still making ATP, and that ATP can be used to convert creatine to creatine phosphate and glucose to glycogen and sort of store that excess energy. When you start moving around, so moderate activities, the ATP demand increases, and we rely on aerobic metabolism, aerobic respiration, to make ATP. There's more oxygen, so there's no fatigue until you are run out of your reserves here. At peak, so you're really working it, really breaking a sweat, that's a lot of ATP demands, and so this is where we might go anaerobic. At maximum produces about 1/3 of the ATP needed, those mitochondria. So that anaerobic fermentation is lactic acid fermentation. So we actually, glycolysis occurs, but that pyruvate is turned into lactate. And that lactate or lactic acid increases the H+, drops the pH, which is called lactic acidosis. And that's what causes muscle fatigue. So that lactic acid buildup slows us down. Nothing to worry about. The lactic acid is recycled in our liver. So as soon as we slow down our peak activity and start breathing more oxygen, those mitochondria can go back to aerobic respiration. All right, that's it for this one. Short and sweet. The last one, we'll cover the rest of the chapter.